Well, welcome everyone. My name is Malcolm Reed. I was professor of philosophy at this esteemed institution for 40 years, retiring in 2007. So why am I introducing people? Well, Bill Barker, who's the director of the Center for Faith and Inquiry, and I serve on that advisory council, asked me to do it. So here I am. Allow me first to say something about the Herrmann lectures, but first I'd very much like to introduce Betty Herrmann, who is able to be here this evening, Bob Herrmann's wife. So unfortunately, Bob is not able, due to ill health, to be with us this evening. Today's lecture and response marks the first of 3 lectures in this year's lecture series. And we would like to begin by sharing with you a little bit about the lecture's namesake, Dr. Robert Herrmann. Bob Herrmann is a former adjunct professor and pre-med advisor at Gordon College. In fact, I think he really got the pre-med program here going well. He also taught medical school biochemistry for 23 years at Boston University and later at Oral Roberts University. In 1981, Dr. Herrmann left medical education and became the executive director of the American Scientific Affiliation, a 2,200-member society of professionals who are interested in integrating Christian faith and the natural sciences. As part of Dr. Herrmann's work with the American Scientific Affiliation, he met Sir John Templeton, with whom Dr. Herrmann collaborated to write several books and also wrote a biography of John Templeton. We are grateful to that very John Templeton Foundation, which serves as the key sponsor and supporter of this Herrmann lectureship series. Now, in a moment, I will introduce our speaker for the lecture series, but first, here's a little overview of how the evening will go. We will hear from our Herrmann lecture series speaker, but first, following this lecture, we'll hear a response from a colleague here at Gordon College, who I'll introduce in a moment. And finally, then we will proceed to a question and answer session, and our speaker and the respondent will stand here, and you can raise your hand and they will acknowledge you and try to answer your question, I'm sure. Thank you. It is now my privilege to introduce the 2019 Herrmann Lecture Series speaker. Dr. Praveen Sethupathy is an associate professor of biomedical science and director of the Center for Genomics at Cornell University, where he directs a research lab focused on genomic approaches to understand human health and disease. He received his BA degree from Cornell and his PhD in genomics from the University of Pennsylvania. After completing a postdoctorate fellowship at the National Human Genome Research Institute under the mentorship of Dr. Francis Collins, he moved in 2011 to the University of North Carolina at Chapel Hill as an assistant professor in the Department of Genomics. The same year, he was selected by Genome Technology as one of the nation's top 25 rising young investigators in genomics. The same year, he was selected by Genome Technology as one of the nation's top 25 rising young investigators in genomics. In 2017, he returned to Cornell as an associate professor, and Praveen has authored over 90 peer-reviewed publications in scientific journals and served as a reviewer in over 35 different journals. Recent honors include the Faculty Merit Award for Outstanding Teaching and Mentoring and the prestigious American Diabetes Association Pathway to Stop Diabetes Research Accelerator, which is awarded to only 3 people a year. Praveen has been an invited speaker for the Veritas Forum and has served on the advisory board of AAAS Dialogues on Science, Ethics, and Religion, and serves on the board of directors of the BioLogos Foundation. That foundation seeks to do work to convey a harmony between science and the Christian faith. He lives in Ithaca, New York, with his wife and 3 children. So a round of applause for our Herrmann lecture speaker.
Thank you very much. Can everybody hear me okay? The most important part of that entire lovely and warm introduction was the last part, my wife and my 3 kids. So get nothing else out of this and get that out of it. Thank you very much to the Center for Faith and Inquiry for inviting me, and to all of Gordon College for so warmly hosting me. It's been a wonderful first day of my visit, so thank you very much to all of you. I'd also like to make sure that I give a special thanks to Hannah Craig. There she is, who has done most of the heavy lifting, planning for this visit and dealing with the tardiness of my responses and all of that. So really appreciate that, and everyone's efforts for making this week happen. Thanks to all of you for coming. I know there are probably many other things that you could do with your late afternoon, evening time. Thanks for spending some of it here. I hope we have some productive time together. I'm really genuinely honored to be the speaker for this year's Robert Herrmann Lecture Series. When Dr. Barker reached out to me and invited me for this particular lecture series, it gave me an opportunity to learn more about Bob Herrmann. And I was thoroughly impressed with the kind of impact that he has had in this community and well beyond, with his relationship with Sir John Templeton and through many other means. So it's really an honor for me. I'm very happy that the family could be in attendance today, and I thank you very much for being here. He was not only a devoted educator and teacher, as you just heard, but also a man that was deeply committed to promoting thoughtful discourse at the intersection of science and faith. There's a lot of noise at that intersection, but he really promoted a thoughtful discourse. And that is of tremendous importance and remains so to this day. So it's really a privilege for me to stand before you as the Herrmann lecturer today. And I really hope that I, today and in the series to follow, can contribute to the wonderful ongoing dialogue that has already been fostered by this series on campus. Today's lecture is about the new frontiers in biomedical research. I thought what I would do today is to walk you through a couple of really exciting areas in biomedicine that my lab is involved in, but many other labs as well. But before I dive into that, I thought it would be best if first I share a little bit about who I am, so you know who you're listening to, okay? So I'm a Christian. I'm also a scientist with a Hindu background, Indian ancestry, Canadian birth, and American citizenship. So that is a lot to unpack, and obviously I won't be getting into most of that today. But what is most relevant for today's lecture and conversation is that as a scientist, I am firmly committed to the pursuit of empirical knowledge, as it should be, making observations and gathering tangible data to test my hypotheses. On the other hand, I am also a person of deep faith in Christ and in truths that I believe science alone cannot illuminate. I've committed my life to both science and faith. They don't occupy separate parts of my brain, and as far as I'm aware, they don't represent multiple personalities. They really do seem to harmoniously commingle as partners. And I'm often asked about why I endeavor to talk about the topic of science and faith in the academy with students and faculty, as well as in the church with fellow brothers and sisters in Christ. And there are many reasons, but in the interest of time, I thought it would be constructive if I started today by sharing with you just one of these reasons. And that reason is that, simply put, it is a difficult, challenging topic. And I believe that makes both academia and the church extremely appealing forums. It's not always the case, but I believe it should be. A shared theme between my experiences as a scientist and as a follower of Christ is that I never stop learning. How many of us have opened up the Bible to a very familiar passage, one that we've read hundreds, thousands of times perhaps, but because of a new context in our lives or something new and fresh that God's been revealing, there's suddenly a fresh new meaning to the words. It doesn't take away what it meant before, but there's another layer that's been added to our understanding of Scripture. The same has been true for me in science. We've had the human genome sequenced now for almost 20 years, and I'll be speaking more about that tonight. And I've personally stared at parts of these genomes for years at a time. And just when I think I've figured out how it works, I'll get a new clue, and I'll learn that it is far more complicated, far more multifunctional than I ever originally imagined. The lifeblood of both vibrant science and vibrant faith is the willingness to wrestle with hard questions that don't have pat answers. But this was not my experience when I was asking difficult questions at the interface of science and faith as a graduate student at Penn. I couldn't find very many partners. They either weren't there or they were in the closet at either my university or my church. I believe we should be a little concerned when the academy and the church get a little too comfortable, too relaxed in its ways, because I think this holds us back from making progress in science and from drawing closer to God, which really is the end for us, is it not? It can lead to a kind of groupthink mentality that can be stagnating and unproductive. I believe that the Center for Faith and Inquiry here at Gordon is committed to, even characterized by, this principle. And it's one of the many reasons that I'm really genuinely honored to be here today as the center's guest. Historically, science and faith have actually been co-laborers in the pursuit of truth. And some of the greatest thinkers, greatest intellects, greatest inspirations to many of us in the audience were profoundly committed to the idea of harmony between science and faith. One of my favorite examples, my favorite scientists, Johannes Kepler. He didn't get into science and stumble in and then just sort of hope that science and faith could coexist. He was driven to do science because of an insatiable desire to learn about God and then to celebrate Him. This modern notion that we hear about a lot, that deep faith in what cannot be seen, is somehow in direct opposition to one's ability to do rigorous study of what can be seen, is just a fallacy. So, a brief overview of my scientific trajectory before we get into some really exciting areas of biomedicine. As was mentioned in the introduction, I did do my bachelor's degree at Cornell University, but it was in computer science. For various reasons, and if you're interested, I'm happy to address them during Q&A, I switched to a more biologically oriented field, but one in which I could apply the skills that I had gained from my studies in computer science. Right? So I turned to computational biology and genomics, and I received my PhD in that at the University of Pennsylvania. Having been trained in both experimental and computational genomics, I felt that for my next step, for my postdoctoral fellowship, I wanted to apply those skills to a human disease setting. I wanted to better understand human disease and contribute to the development of new therapeutic approaches for patients who are suffering. That's what I did during my postdoctoral studies when I trained with Dr. Collins at the NIH, and then I moved on to set up my lab at UNC Chapel Hill. Because of the nature of my training, I had the privilege of being able to set up a lab that was both computational and experimental. So right from the get-go, my training and my professional journey have been about collaboration across many different disciplines. I simply don't know any other way to do science. Right? It's just a part of my DNA, if you will. Sorry, no pun. Along the way, I of course asked myself many times, and this particular portion here is directed to any undergrads in the audience who may be feeling the way that I was feeling as a senior, thinking about my future, wondering what I should be doing. I was a new Christian. I had become a Christian during my college years at Cornell. And I joined in with my colleagues in this paralyzing, crippling fear of what I was supposed to do with my life. Finding God's will turned out not to be as much of a joy as I thought it might be, and it was rather paralyzing. Right? because I had this fallacious notion that here was the opportunity I had to make the right decision, and if I missed the boat, that's it, I just couldn't honor God for the rest of my life. But of course, thankfully, I learned that it's not so much what I do, but why I do it and how I do it that really is what ultimately God cares about. So I'm sure he's pleased that I'm a scientist, but I think he's much more interested in how I'm a scientist and why I'm a scientist, my motivation and my attitude. And I share that because it was very freeing for me, and I hope it's freeing for you if you're going through something similar. There's freedom in Christ about what we do, right? But I encourage you to pay attention to your heart and your motivation, your posture toward that chosen craft. Why are you doing it, and how are you doing it? With that, let me get into the meat of today's lecture, and that is to talk about 3 different topics within the area of biomedicine that are particularly exciting and we're at a sort of new frontier on. These 3 topics are exploring the dark matter of the genome, that's number 1. Number 2, working toward precision medicine. And number 3, the world of epigenetics. Okay? If these terms are already deeply familiar to some of you, I'm still hoping very much that you'll get something useful out of my lecture. If they're not familiar to you, don't worry. We're going to walk through them step by step. Okay? Okay, dark matter of the genome. The first draft of the human genome, this is the full sequence of human DNA, was completed at the turn of the millennium. So there are students at Gordon today, right, who weren't alive when we didn't know the human genome, right, which is astounding for me to think about. This was less than just 20 years ago. And when the announcement was made from the White House, President Bill Clinton referred to the genome as the most important, most wondrous map ever produced by humankind. That day, there was certainly an incredible sense of awe at what we had accomplished. But I felt, mixed in, was a tremendous sense of hope. And I think it's because the true beauty of a remarkable achievement lies not necessarily in the past work that was done to accomplish it, but in the fresh and new ways that it stirs our imagination to carve a better, more healthy future for humanity. I think it was in the possibilities of having the human genome that we were even more excited than the accomplishment of having done it. The sequencing of the human genome unveiled what we like to call the instruction booklet for every cell in our body, nearly every cell in our body. Truly amazing indeed. But when the dust settled on the discovery, we realized we did not know how to interpret the instructions because the language in which it was written was largely unknown to us. What was actually encoded in the genome? How does it work? What does it teach us about our bodies or why we get diseases? These were all things we didn't actually have answers to, but we were hopeful we might. And that brings us to this first topic, which is the dark matter of the genome. Many of you will be more familiar with the concept of dark matter in astrophysics, right? It's material that is not comprised of atoms as we've defined it, not directly observable with today's methodologies, but nonetheless absolutely required, right? Their gravitational effects are required for the large-scale structure of the universe. Dark matter constitutes much, much more of the universe than all of what we call normal matter, and yet we don't actually know what it's comprised of. If you were to talk to a physicist, he or she would be able to tell you more than I could about ideas and speculations in the field about what it's comprised of, but this is still very much an ongoing investigation. We're in the dark, so to speak, about what it is. It turns out that there's an analogous concept with the human genome. The genome is the full sequence of our DNA. It's the hereditary material that's passed on from one generation to the next. You can think of DNA as a language, right, that has only 4 letters in its alphabet. And you can string these 4 letters in various orders, right, and get a book that's 3 billion letters long. That would be your genome, right? And my version of that book would be ever so slightly different from yours, okay? A 3 billion letter long book. That's essentially what it is. And we tend to think of the book as a set of instructions, which in the field we refer to as genes, and you will have likely heard of that term as well. The word genome actually literally means a compendium or collection of genes. And later on, I'll talk about a gene getting turned on or off. You can think about that basically as an instruction getting executed or not executed. Okay? So that's what gene activation is. But it turns out that the genome is much more than just genes. In fact, the parts of the genes that get interpreted as the instructions for our cells to execute their functions only constitute about 1% to 2% of the genome. What is all the rest of it? Right? Has been a question that we have been asking for at least 20 years, and actually more, even before the genome era. We basically didn't know, and so we referred to it as dark matter, taking the cue from our astrophysics friends. But over the last 2 decades, the dark matter has slowly but steadily been coming to light. And here's now what we see. Much of what we call dark matter in the genome is filled with little DNA rheostats, or dials. You know those light rheostats sometimes we have in our houses where it's just dialing it up or down the light in a room? You can think of a bulk of the rest of the genome being littered with these kinds of rheostats, okay? And what are they doing? What are they rheostating? Right? It turns out that they're controlling when, where, and how much nearby genes are being turned on, right? So when we talk about genes being in the genome, when they're just in our DNA, they're static. They're not actually doing anything. They need to be activated in order for that gene to exert its function, right? That's what we call executing that instruction. Well, how does the cell know when to activate it, how much to activate it, right? It turns out that information is encoded in these little rheostats that are littered throughout the DNA. Right? We knew that there were these rheostats in the genome. It's not as though the concept was new to us, right? But just how many different kinds of rheostats there are and just how much of the genome they occupy was rather startling to us. As an example, my lab recently discovered a particular rheostat encoded in the DNA that turns out it's controlling the activation of genes that are responsible for producing cholesterol in our liver. Right? When the rheostat isn't working properly, let's say it's dialed up a little too high, then the liver might be making too much cholesterol, irrespective of how much cholesterol we're taking in by diet, and that increases our risk for fatty liver disease, heart disease, a host of other clinical problems. Right? So, having discovered that there were these rheostats littered across the genome, the next major challenge in the genomics field was figuring out exactly where all the rheostats were and which ones were functioning in which parts of the body. Right? They're not all functioning all the time. Some are more relevant in the brain, some are more relevant in the pancreas, and so on. Right? And we didn't have a sense for that. And so a national effort was initiated by the National Institutes of Health, and it was a project called Encode, an extraordinarily ambitious effort. Encode stands for Encyclopedia of DNA Elements. Right? So the goal was really to discover where all of these rheostats were across the entire genome and hopefully figure out which ones were relevant in what parts of the body. Okay. It's turned out over the last 15 years of this particular project that it's tremendously advanced our understanding of how the genome works, but it's also transformed our understanding of human disease. Here's the punchline. What we've learned in the last 15 years is that genetic mutations that are known to increase your risk for a variety of different complex diseases, whether it be neurological conditions or cardiovascular conditions or metabolic conditions, rather than occurring in the genes themselves, they much more frequently occur in the rheostats. Now, this doesn't mean that what you learned in molecular biology class, that you can have genes with mutations that increase your risk for disease, is false. Of course that's true, and that can happen, and you may know about some of these examples, like BRCA1 in breast cancer, for example, that Angelina Jolie really popularized a few years ago. No doubt those exist, but it turns out the vast majority of mutations that confer risk for complex diseases are not in the genes themselves, but likely in these rheostats or these dials. Right? And so that puts us in an era today where we are thinking about ways in which we can develop therapeutic modalities to target these rheostats and not necessarily the genes that we've been focused on for many, many decades prior. Right? So it's a complete paradigm shift. We used to think about disease as qualitative. You have the bad gene for this or the defective gene for that. Right? While that can happen, we're increasingly realizing that most complex diseases are better described as your gene is fine. You should be turning it on at 10 units and you're turning it on at 100 instead. Or you should be turning it on at 200 units and you're only got it dialed up to 40, right? It turns out diseases are more quantitative, right, than we previously gave credit. And that has been a major paradigm shift, and it's changed the way that we think about pharmacological therapy for patients who are suffering. That brings me to the second main topic, and that's precision medicine. Can I have a show of hands? How many people have actually heard that term, precision medicine? Okay, so a quarter maybe, right? Since the dawn of the genome era at the turn of the millennium, like we talked about, there's been talk of this notion of personalized genomics. That's another term that you may have heard about. I'm seeing some nodding heads. Right? And the idea here is that an individual's genome, yours or mine, right, can provide detailed information on the precise treatment strategy that a clinician may recommend. Right? This is what is also now more popularly referred to as precision medicine. It's about being more precise in the medications that are offered. And for a long time, this seemed like a really wonderful idea, but basically impossible. Right? But in recent years, we're not only applying it in the clinic, but it is seeming like it is increasingly within reach for far more diseases than are currently being leveraged for. A lot of this has to do with the fact that there have been tremendous advances in technology that have made it faster and cheaper to sequence your human genome. So I want to give you an idea of how much faster and cheaper it's gotten. In the year 2000, again, the turn of the millennium, when the first draft of the human genome was published, does anybody have a guess as to how much it cost to sequence the human genome at that time? Yeah, actually, some estimates, they range, but $50 million is one estimate that has been put out there. Okay. 10 years later, in 2010, the cost was an order of magnitude less, around $5 million. So that sounds great, but I'm pretty sure that you or I don't have $5 million to burn if we want to get our genome sequenced, right? If you do, let me know. I want to make friends. Today, another 10 years later, it's about $1,000. And we're starting to get somewhere, right? And it's getting cheaper by the day. Moreover, the first human genome sequence took 10 years to complete. This is why we went to the White House to announce it. Today, if you want your genome sequenced, you can do it overnight. This has happened in less than a generation. It's happened in 20 years. Truly staggering and remarkable to think about that kind of pace in the development of technology. Sir William Osler, I don't know if you'll be familiar with the name, is a famous Canadian physician and a co-founder, actually, of Johns Hopkins Hospital. He said, in the late 1800s, if it were not for the great variability among individuals, medicine might as well be a science. But unfortunately, it's more of an art. Right? This clever quote reflects the common frustration, even today, that medicine is not as precise as it needs to be. Traditionally, most therapies, we're all familiar with this when we go to the doctor. Most therapies are applied to the so-called statistically average patient. Right? But what if we could tailor medical treatment to each specific individual based on their unique characteristics, including their unique genome. This seemed kind of like a pipe dream, although worth talking about at least 20, 25 years ago. But like I said, today it's becoming a reality. There's a long way still to go for this to be widely leveraged, but it is becoming a reality. And I want to tell you about a couple of really exciting examples of this. So earlier in the talk, I talked about genes getting turned on. Well, it turns out there are about 25,000 genes encoded in your genome, the 1% to 2% we talked about. And of those 25,000 genes, a small set might be on and the others will be off. So let's call that the on-off profile of any given cell, okay? Every cell in the body actually has a slightly different on-off profile, and that's what leads to their slightly different functions. And the brain cell better have a completely different on-off profile than the pancreas cell because we need it to be doing something totally different, right? Well, it's not only the case that the on-off profile is different among cells in a healthy body, but it turns out that if I were to take healthy tissue and cancerous tissue, I would notice that the on-off profiles are completely and reproducibly different in the cancer tissue compared to the normal tissue. This was first examined and reported in breast cancer. So there was normal breast tissue and breast tumors that were analyzed. And at first, it was evident that the on-off profiles were completely different in the tumor compared to the healthy tissue. But then further investigation, what the scientists realized is that if you just focused on the tumor samples, you could actually detect 4 different clusters within the tumor samples, 4 different on-off patterns. Not every breast tumor from a patient exhibited the same dysfunctional on-off pattern. There were at least 4 different on-off patterns. This led to the discovery of at least 4 different subtypes of breast cancer. Up until then, it had been postulated that there must be different subtypes because not everybody responded to the same medications with the same level of efficacy. But it wasn't really understood why. And what this molecular work did, the ability to peer into a cell and ask this remarkable question of which genes are on and which genes are off, and build an on-off pattern, allowed us to be able to see there are at least 4 on-off patterns, meaning there are at least 4 different subtypes of breast cancer, and it turns out each of those subtypes respond very differently to medication. Oh my gosh, how was that revolutionary? Because now you could go into the clinic, figure out what subtype you had based on the on-off pattern, and get the appropriate medication rather than wasting time going through a litany of other medications. Right? So it alleviated quite a bit of suffering for patients and actually put people in remission more effectively than previously. Right? We didn't solve breast cancer, okay? But there was a tremendous advance because of genomic technology, because of the fact that we knew what these genes were and had the technology available to us to actually measure the on-offness of those genes. This has now been followed up in a number of other cancers: lung cancer, colon cancer, melanoma, leukemia. They're all following suit and being able to apply this kind of methodology to improve their treatment regimen for patients. Another really compelling example, many of you may know about warfarin, blood thinner. Right? It's applied, prescribed for individuals who have blood clots, prevents the risk of stroke and other kind of downstream complications. Okay, widely prescribed medication. Right? But it was very frustrating that some people would respond to what we thought was the appropriate dose of warfarin better than others. Right? But with the advent of being able to sequence people's genomes and some downstream studies that were done, what was realized is that there is a specific mutation, this time actually in the gene, not in a rheostat, but there is a specific mutation that if you had that mutation in a particular gene, you would actually require a much higher dose. Because it turned out that this gene, when activated, was an enzyme, and was an enzyme that metabolized the drug. So it broke it down really effectively if you had that mutation. So what we can now do, you come into the clinic, we can screen you for that mutation, and if you have that mutation, we'll probably start off with a much higher dose of warfarin instead of wasting time with the lower doses that we thought were more appropriate. Right? These are just a handful of examples of ways in which we are getting closer to this idea of precision medicine. Final topic that I want to address, you guys still with me? Not too much science? All right. Is the concept of epigenetics. So earlier I referred to the genome as a set of instructions, and every cell in the body more or less has those same instructions, the same DNA. So if that's the case, how is it that the brain cell knows to do something very different from the pancreas cell? They have the same instructions, I just finished saying it, right? So how is it that they're actually able to do different things even though they're executing the same instructions? It turns out that the cells in our body are programmed to know which instructions to execute and which ones to ignore. So let's say that we've got instructions A to Z. Somehow the brain cell, the neuron, knows I only ought to do A, B, and C, and that'll allow me to function like a brain cell. Whereas in the pancreas it says, yes, I see A to Z, but I know I'm only really supposed to do X, Y, and Z, and that allows me to function like a proper pancreatic cell. How could that be? Right? And I could spend the next several days talking with you about the kinds of hypotheses that we're pursuing and the ideas that we have. We don't actually have a definitive answer. I can talk around it quite a bit and give you a whole course in epigenetics, but you'd still walk away going, okay, so we know a lot, but we still don't know exactly how that happens, right? I mean, if you stretched out your DNA, let's just talk about DNA for a minute. If you stretched out your DNA just from one cell of your body, from end to end, it'd be about 6-7 feet. If you took all the cells in your body and took the DNA out and laid them out end to end, stretching them out, it'd be hundreds of millions of miles, right? That much DNA is packed into one body. I get goosebumps, I know this, but I get goosebumps saying it every time I say it because it's a remarkable amount of packaging. Look at how much information that is, and it's crammed into this tiny, tiny, tiny little space. Right? In order to be crammed into such a space, right, it requires a very robust packaging material. Turns out, DNA is not naked in the nucleus of your cells. It is actually wrapped up in this very special packaging material. We refer to it as chromatin, but the names are not that important. The important point is that marks can be made on the packaging material. You can think of it as you just making a mark with your pen, but it's really the cell adding a little chemical group to certain parts of the packaging material. But again, we can abstract it out and just talk about it as marks that can be made, right? And there are hundreds of different kinds of marks. Combinations of different marks turn out to be a communication to the cell about whether that region is an instruction to be executed or not. So we'll talk about it as open or closed, or activated or silenced, right? Remember the brain had A, B, and C open and everything else was closed? Well, that is because the A, B, and C instructions were packaged in a particular location where marks were made that told the cell, this is available for you to execute. Whereas the rest of it, those marks weren't there, right? And the same for the pancreas cell and so on. Right? But really the remarkable thing about this, if that wasn't remarkable enough, is that the environment around us, our lifestyle choices, our environmental exposures, our diet, whether we exercise or not, everything that we do or don't do, they can actually make modifications to the packaging material. And you now know that such modifications to the packaging material can make instructions either accessible or not accessible to the cell. And so you can essentially modify how your cell is behaving without affecting the underlying DNA at all. All you've done is through your choices and through your environment, modified the packaging material so that some regions are on and some regions are off. This is essentially in the genetics arena how we think about cancer. Basically, cancer is a complete rewiring of the on-off pattern, right? As I talked about with breast cancer. And it's in part, not completely, because of how environmental influences, right, can alter that on-off pattern in the genome of a particular cell. Right? Not only that, right, but there is now good evidence that those kinds of marks, some of them can be heritable in plants, in worms, and to some extent in mice. The extent to which this happens in mammals and humans, I want to stress, is unclear. The conventional wisdom still states it's probably not happening very much, and I think we're not going to cross the hurdle of accepting that it does, should that be the case, until we can really delineate the mechanisms by which it is doing so. Okay? So, all of those caveats issued, is still an extraordinarily exciting arena of investigation right now. Not only can we pass on our genetic material, our DNA, as we're well aware of, right? Could it possibly be that we are passing on to our children also a history of the environments that we've been exposed to, right? This is, I want to emphasize, you'll hear things like that, and I want to be clear that that is a bit sensationalized with respect to where we are in the science right now. So I hope that's very clear to everybody. But I just want you to be aware of the fact that that is a conversation that is happening right now, and there is rigorous science that is being done to address that question. So I hope to come back, and maybe in another decade, I'll have more that I can say about that, right? But if you're curious and you want to look up more about it, it's called Transgenerational Inheritance, Transgenerational Epigenetic Inheritance, okay? And there's no doubt you will read quite a bit about how it's controversial and how we, for various good reasons, believe that it's probably not happening very much. But even if it was happening a little bit, it would be really extraordinary discovery. There are many other topics that I could have chosen to tell you about today. Neuroscience is probably the area that is exploding more than anything else right now. It's not as much in my wheelhouse, so I stayed away from it, but I've got a sister who's a neuroscientist at Rockefeller University, and I glean from her all of the exciting work that's going on in that field. We're finally getting to the point where we're understanding at the molecular level how memories are made, right? How that could be leveraged for being able to help Alzheimer's patients or people with PTSD on the other side of the spectrum, right? So just really, really fascinating stuff. There's CRISPR-Cas9 genome editing, which I'm sure many of you have heard about. We'll talk about that tomorrow. There's the microbiome, right, which I will briefly mention in my lecture on Wednesday, and many, many more topics. Okay, but what I want to leave you with today is, okay, there are all these really exciting things going on in the biomedical space. This is a lecture series on science and faith. Right? What, if anything, has all of this, the work in these incredibly exciting arenas, what has that taught me? What kind of spiritual grounding, if anything, has that given me? How has it influenced my faith, if at all? Again, there's a lot that I could say about that, but I'll leave you with three things. Aren't like good sermons supposed to have three things? So the three things are collaboration, humility, and worship. None of what I talked about today would have been even remotely possible had it not been for incredibly large-scale, consistent, and sustained collaboration across many different units in this country and world. And not only that, not only a geographic collaboration that's impressive, but across physics, chemistry, biology, medicine, computer science, mathematics, the list goes on. You needed people across all of those disciplines to have a buy-in to see the value of this effort and then come together to work. One of those or two of those weren't there, we would not be where we are today. We just wouldn't. Okay? In my work as a molecular biologist and as a geneticist, I'm leveraging technologies thanks to advances in physics and chemistry that I don't fully understand. Right? So this is really a remarkable testament to the importance of cooperation and collaboration to really accomplish anything. And that gets messy. Where there are people, it will be messy. But it's how we move forward. And I think as Christians, we should embrace that messy reality and say we've got to work together if we're going to accomplish anything done that's meaningful. The second is humility. And for that, I can barely do better than a quote from Johannes Kepler, so I will just read it out. when someone this was sort of in response to the greatness of his discoveries, he said, "I was merely thinking God's thoughts after him. Since we astronomers are priests of the highest God in regard to the book of nature, it benefits us to be thoughtful, not of the glory of our minds, but rather, above all else, of the glory of God." Right? There's so much in there that's really beautiful, right? He thinks of himself as a high priest of God, as someone who is exploring nature. I wish we presented it that way to our budding scientists in our Christian communities across our country, right? That you're a high priest of God of the book of nature that he authored, that he created. And finally, worship. I'll say this again at the Wednesday lecture, but the work that I do, it reminds me on a regular basis that we are that slice of creation that gets to delight in creation alongside our Creator. We tend to, as biologists, we tend to focus a lot on those aspects and features that we have shared with many other species. And it is really remarkable to consider that. But consider that we are that unique slice of creation that is delighting in creation alongside our Creator. For the believer, inquiry in the natural world should lead to awe. Psalm 19 kind of awe. O Lord my God, when I, in awesome wonder, consider all the worlds thy hands have made. Thank you.
Thank you, Praveen. So masterfully presented. Thank you so much. Thank you. It's my joy now to introduce a faculty member who was not here when I was teaching here, but delighted to have met her prior to this gathering. Dr. Angie Cornwell joined the Gordon College faculty in 2014, so fairly recently. Her primary research involves understanding molecular signaling pathways leading to airway or gut remodeling in allergic diseases such as inflammatory asthma and eosinophil-driven gastrointestinal disorders. Now, if you understand that, I'll talk to you later. It's wonderful, though. She was selected to participate in the fellowship program, Bridging the Two Cultures of Science and Humanities, Part 2, sponsored by SCIO, or Scholarship and Christianity in Oxford. Her work in the SCIO program focuses on the integration of faith and science, particularly the uniquely Christian perspective on recent developments in medical biotechnology. She serves as an internship coordinator, liaison to the North Shore Technology Council, and as an advisor to the North Shore Student Chapter of the American Scientific Affiliation. So please join me in welcoming now to this stage Dr. Angie Cornwell.
Thank you. Dr. Sethupathy, thank you for that very passionate and energetic talk that we just heard, and thank you for agreeing to come to Gordon College for these 3 days for the Herrmann lectures. And thank you to Mrs. Herrmann and your husband for the support of these important lectures and an important area where we can come together and have thoughtful dialogue. We're honored to have you. I'm honored to be here, honored to have you here with us, Dr. Sethupathy. And we are so thrilled to hear about your work on DNA rheostats and on epigenetic modifications and all the things that you've been doing, exciting projects that you've been doing, trying to understand the dark matter of the human genome and what's going on there. sometimes when my students hear about these discoveries, they become intimidated, and they think that there's nothing that they can contribute to this world of discovery. They're intimidated by how thick their textbooks are, how much knowledge has come before them, the people who have come before them that they're learning about, and they think, what can I contribute in this world? And so we are excited to learn that there's still so much yet to be discovered, and that we've only just touched the tip of the iceberg in terms of our understanding of the human genome and how it's been being regulated. So this talk is partly to share how much we have yet to do, but also for the non-scientists in the room to share what you can contribute as well, because we all need to be part of this dialogue. And so part of this Herrmann lecture series to stimulate thought-provoking discussion this is a very important job that we all have, scientists and non-scientists alike. So we've been really privileged to be in this time and place in science. And in the past 2 decades, uncovering the human genome, the sequencing of the human genome, and learning about the importance of all these post-transcriptional gene silencing, really important. But there are several points that I would like to make to the audience, to this mixed audience. First, while this is a really, truly exciting time in biomedical research, and there have been so many advances, some of which Dr. Sethupathy shared with us, others that we have not heard about in this forum, but including things like regeneration of the sciatic nerve, regeneration of the retina, things involving bioengineering, regeneration of pancreatic function with transplant of beta cells and pancreatic islets, growth of miniature organs in dishes. All of these things are really exciting so that we could try to work on transplanting these types of organs. We're not there yet, but we're working on developing these technologies. In addition, we've had major breakthroughs in using human cells in these sort of lung on chip and other organs on a chip, where we can get away from the animal models, where we know that there are gaps in what the animal models can provide us with human models. And so we can use these lung on a chip, organ on a chip, to study interactions in human cells without having to do experimentation on humans. So all of these are really exciting developments in biomedicine. Other kinds of developments include using light-sensitive ion channels, where we can use pulses of light to study in seconds of time, where we can study the effects of these micropulses of light in opening and closing ion channels. So there's much to be excited about in the field of biomedicine, and much that we can learn about regulation of neuron function, of skeletal muscle and cardiac muscle. In addition, there are other more perhaps controversial types of biomedical discoveries, such as 3-person in vitro fertilization, where women with mitochondrial defects can now give birth to babies that are related to them, biologically related to them by nuclear DNA, but where the mitochondrial DNA comes from a third donor. So some of these are a little bit controversial, but these technologies are available. And of course, we look forward to tomorrow's comments on CRISPR-Cas9 and a whole wealth of possibilities with CRISPR-Cas9, not only with gene editing, but also with transcriptional regulation, where we can turn genes on and off, we can modify them and so forth using CRISPR-Cas technology. So a lot of really exciting developments with potential for treatment in HIV, for things like congenital blindness, hemophilia, sickle cell disease, all kinds of disorders and all kinds of cancers. These are very exciting times. Gordon College is placed in a very special location to be able to take advantage of these developments in science, and especially here on the beautiful North Shore of Massachusetts, we are in a hub that is rapidly becoming the Silicon Valley of biotechnology. And so our state deliberately set aside money for the purpose of growing biotechnology, especially in the greater Boston area. At Gordon, we, along with a handful of other schools, in 2008, received significant funding, capital funding, to improve our infrastructure, to improve our equipment and our space. The cadaver lab was part of that improvement. And in 2008, as part of this funding, the Massachusetts Life Sciences Center was charged with allocating $1 billion in funding to support promising startup companies and to stimulate collaborations between academia and industry. So here on the North Shore, we're in a particular place of privilege to be able to take advantage of these developments in biotechnology and in medicine, biomedicine. In addition, our access to places like hospitals in the Longwood Medical Center, to places like companies that are centered here in Cambridge and Greater Boston and even north of Greater Boston, our students have been able to take advantage of wonderful opportunities to participate in internships in some of the finest hospitals in the world, to participate in internships at some of the finest companies in the world, providing equipment and supplies for biomedical research. So we're positioned in a very special location here at Gordon College. And so students at Gordon have also been taking advantage of the opportunity to hear lectures and attend conferences that are available to us because we're in such proximity to some of these wonderful places like Harvard Medical Center and so forth. So just on Friday of last week, 20 of the Gordon students went and attended a lecture by Brock Reeve, who is the executive director of the Harvard Stem Cell Institute and CEO of a company called Iviva Medical, and they heard about his company's work designing biological platforms to treat renal disease. So making a biological platform with channels that could then be seeded with cells that would then allow us to study renal function and renal disease. So a lot of incredibly exciting programming as part of the North Shore Technology Council organization of talks of special First Friday Biosciences lectures. We also have had the ability to travel just 10 minutes up the road to the Gloucester Marine Genomics Institute, where our class heard Dr. Andrea Bodner speak about sea urchins, the longevity of sea urchins, and the applications to the study of human aging. So we have some really groundbreaking research going on. In addition, there are researchers studying environmental DNA and looking at applications for human health and the health of the planet. And so we've been able to take advantage of talks at the Harvard Museum of Natural History, including talks on ancient DNA and the science of our human past. We've been able to hear researchers talk about genes, cognition, and as Dr. Sethupathy alluded, in the field of neuroscience developments, really exciting developments there. We've been able to take advantage of conferences that have come to the Boston area where students can attend these conferences without charge and hear some of the most prominent researchers in the field. So we are positioned in a place where we can have wonderful opportunities to hear amazing lecturers. And so, as Dr. Sethupathy said, never stop learning. We're in an environment where we have the ability to hear so many wonderful things and to increase and deepen our understanding. My third point is that with this scientific knowledge comes great responsibility. And so we have the responsibility, especially as Christians in science, we have the responsibility to use our knowledge wisely and judiciously. We have the responsibility as stewards of the planet to protect the resources that we have on the planet and to protect those with whom we are in community. And by those with whom we are in community, I mean not only our fellow persons on the planet, but also the fellow animals and plants and even bacteria, I suppose, that share the planet with us. So we are called not to squander those resources that we've been given, not to waste our talents, but to use our wisdom, whatever we can learn from our scientific discovery, to use these in a way that will benefit the planet and those that are on the planet. We have also the responsibility to protect those who are poor and helpless among us. So not only to cater to the richest in our world, but also to look after the poor, the homeless. And so what should we do? And in this, I'm going to suggest that not only the scientists in this room should be listening for ways that they can contribute to scientific discovery, but everyone in this room can contribute by becoming educated and by seeking ways that they can be part of the discussion, things that are happening in the scientific community. So what are some of the questions that I think we should really be asking ourselves? One, how can we take better care of our neighbors who are here on the planet with us? How do we make sure that the less wealthy among us are not left behind in this great discovery that is coming? How do we give them access to things like personalized medicine, precision medicine? How do we do this when we know that some of the poorest people on the planet still do not have reliable access to clean drinking water, do not have food security, do not know where their next meal will be coming from, and do not have access to basic medical treatment, things that we take for granted, things like just going for annual checkups and routine screenings, things like access to vaccines and medicine. How do we protect those who are less disadvantaged among us? And how do we protect them from diseases that in many countries, these diseases have been eliminated or are no longer a serious concern, but are still a serious health concern in some places in the world? So that's one question. How can we do a better job of protecting those who are not as fortunate as we are in this country? Secondly, how can we help regulatory agencies, perhaps the government or other regulatory bodies, how can we help them to try to protect the most vulnerable among us? In our not so distant past, experiments such as vaccine testing and the characterization of disease progression of some not very nice diseases were carried out on groups of people that we considered to be the marginalized. And I'm talking about prisoners and orphans and people of minority races and patients who were admitted to psychiatric institutions, insane asylums. In the US, we might believe that in our present day, we're beyond these types of practices, that these were injustices in the past. We might not be able to do anything about them, but we can't do anything, we can't worry about them in the present because they're not part of our culture. If we believed ourselves to be free of these kinds of concerns, I would caution us to think about some of these cutting-edge technologies, CRISPR-Cas9, and the implications that some of these have on our researchers. Some of our researchers are traveling to countries where CRISPR-Cas9 on humans, in particular in the human germline, is not regulated. And so this opens up the possibility that volunteers in some of these places where CRISPR-Cas9 is not regulated could be used on people who really don't understand fully the dangers because they're not educated to the same level that we might be in some of in this part of the world. And so they might be pressured into participating in some of these studies because they might be given some small financial incentives to participate. Financial incentives that for a person in this country might not cause them to want to participate, but in a country where many people are of lower socioeconomic status, they might be driven to participate in some of these studies. And so the risks that they might be undertaking are not fully understood. Let us consider the case of thalidomide, a drug that was prescribed to treat nausea in pregnant women. And so many of you have heard of this drug and might think of it as something from the 1960s that we no longer have responsibility for. In 1961, with the growing evidence that mothers being prescribed the drug were giving birth to babies with severe birth defects, the drug was taken off the market in many places, but not everywhere. In Spain, the drug continued to be prescribed for an additional 4 to 5 years, resulting in the birth of an additional 3,000 babies born with thalidomide-related defects, birth defects. And if we realize that in the present day, only 23 of those 3,000 babies that were born ever received any compensation for their disabilities, and furthermore, that it took a full 45 years for those 23 victims to receive compensation. And many of the victims were denied coverage because their term had expired, their term to pursue benefits had expired. This is a warning to us, especially in an age where some of the modifications we may make, especially using CRISPR-Cas9, may have really long-term effects that we cannot predict their outcome very well. So the pharmaceutical company, Grunenthal, which had continued to distribute this drug, this anti-nausea drug, to pregnant women in Spain for 4 to 5 years after the drug was shown to cause birth defects, only had to compensate fewer than 1% of the victims. So we must do better as Christians. We must do better to look after the interests of these vulnerable populations. How can we strategically avoid some harmful outcomes? And this is going to be very difficult because, as Dr. Sethupathy had published in an earlier publication, we do not yet understand long-term effects of many technologies, especially like gene editing. So we must use caution. We must proceed with caution. No matter how good our animal models, they will not be able to predict, they will not be able to close the gap fully between human and animal work. And although we've made great strides in improving seeking after targets and increasing our efficiency, improving our accuracy, we still cannot anticipate all of the outcomes. So there are sure to be errors in humans when these things are carried out, especially in countries where we are not regulating some of the work that's being done. And there are sure to be real victims, human beings, who are suffering as a result of these editing errors. And here again, we find that the legal agencies and regulatory bodies are woefully unprepared to address even the very basic questions about things like gene editing, especially in the human germline. Who is going to be financially responsible? How can we ensure that those who are harmed will receive adequate compensation, even if it's 50 or 100 years later? How many generations would be compensated? Would future generations be barred from reproduction if they are found to have certain edits from undergoing some of these therapies that cause irreparable harm? And in cases where the effects might be very long-term, who will be responsible for even long-term follow-up of the patients? Does an embryo have a right not to be edited? These are all questions that I think we have to consider. And in the case of precision medicine, if we find out that a person has risk factors for disease, then who will have the right to this type of information? Will insurance agencies? Will our employers? If genome editing becomes a common practice in the way vaccines are currently a common practice, or epigenetic genome editing, Will a patient have a choice about whether or not to have things turned on or off? Will they have the freedom to decide for themselves? So while these are very exciting times for discovery in biomedicine, we must also remember that there are serious issues in which all of us can participate by informing ourselves of what science is doing and by participating in the dialogue. And that's why these Herrmann lectures are so important to this discussion. So Dr. Sethupathy, I'll give you a little plug for the next 2 days. The next 2 days of lecture. Tomorrow, we look forward to hearing your remarks on CRISPR-Cas9 genome editing, current applications, and thoughts about ethics of. And we look forward to your third day of discussion on the microbiome and on science and faith. So we thank you for being a part of the Herrmann Lecture Series, and thank you for enlightening us with your thoughts and allowing us to have a platform for further discussion.
I don't need to point to people. I think our two guests can handle you if you just put your hand up and stand up and give a question, and a microphone will be brought to you.
I think we only have time for 2 or 3 questions.
Okay, 2 or 3 questions. Microphone?
Well, I don't see lots of people jumping up, so you mentioned the possibility that environment can change the epigenetic expression of genes. And I'm just wondering what would be a good example in the animal kingdom of maybe an experimental system or a case where the environment can reproducibly do such that maybe people could relate to?
Yeah. So there are really, really good examples in nematodes and plants, but I'll stay away from those because we can probably relate to them less. So with respect to the final part of your question, so I'll give an example from mice, actually, which is actually a newer experiments that have been done, where paternal diet, right, seems to impact the epigenomic on-off profile of the offspring. Right? Why more the paternal than the maternal is an active area of investigation. It gets really complicated when you get into the molecular details of how they believe that that's being transmitted, but they think that they have a mechanism involved as well. But that alteration of the on-off pattern in the offspring because of the diet, and the diets that are usually used in these studies are high-fat diets or high-carbohydrate diets. Many of them are actually referred to as Western diets. And they lead to a change in the on-off pattern in these offspring that they have then not only correlated but causatively connected to an unhealthy change in the metabolic profile of those offspring. Right? So that's probably one of the better studied examples. It's from a researcher, his name's Oliver Rando at the University of Massachusetts, actually. But that's a good example, I think. Anybody else? Chomping at the bit here. Yes.
One thing we hear about sometimes is research aimed at prolonging life. And I'm just curious to hear your thoughts if that's an active or interesting or ethical type of research that you're familiar with.
Yeah. That is really interesting. It's not one, aging-related research is not one that I have committed a lot of my energies toward, primarily because I'm actually more interested in aging gracefully, all right, than I am in prolonging some kind of an aging. So I don't know that there's an ethical problem. I've not been able to really pinpoint a very clear ethical issue here unless there's a specific offshoot of this. Generally speaking, trying to understand what it is that contributes to our aging processes and how we might be able to, in some people, there's accelerated aging. So there's a disease called progeria. people in the aging field may not really like it being called accelerated aging because there are other features of the disease that don't quite track with aging. But it is commonly referred to as such. And so you would see a 10, 12-year-old boy or girl who looks like an 80, 90-year-old person, right? And it's tragic, and they often end up dying prior to their teenage years with cardiovascular events and other kinds of complications. And so if we better understand aging, we might better understand accelerated aging and be able to help patients like those. So I do think there's space and room for it. I do think it's important. But I do also agree that sometimes I do wonder about the disproportionate amount of money we do spend on that line of research versus others.
Hi, thank you for coming to speak to us.
Sure.
With regard to what you said about the ease at which sequencing human genomes is becoming, what can be said about the privacy of such information? Because that's a lot of power over a person knowing their entire genetic makeup.
Yes, fantastic question, and I think goes very much in line with what was shared just now. Francis Collins, whom I mentioned earlier as the director of the NIH, had been on a campaign for many years, basically the bulk of the time that he was NHGRI director and NIH director, to have passed into law what's called GINA, Genetic Information Non-Discrimination Act, GINA. Right? This was really extraordinarily important for exactly the question that you're asking because it restricts certain kinds of agencies and organizations from being able to access that information and discriminate against you on the basis of that information. Right? However, it turns out, I don't remember all of the details, it does not apply to every such possible organization. Right? They had to subset it somehow. Right? So there's now a lot of interest in expanding GINA to GINA 2.0. Right? So that it's not just, let's say, health insurance companies that can't access the information, it's life insurance, right? Which seems like that ought to be important too, right? And other kinds of agencies. Now, in addition to putting a law in place like this, right, we need to have regulatory bodies that are able to audit organizations and make sure that this kind of funny business isn't happening, right? That has been put in place, but the thing is that this is all very new, right? So it's unclear how well it's working. And I think we need more years and more data to really be able to determine that. But these are really the first steps, is putting laws in place, regulating the kind of behavior that's allowed, right? Figuring out the regulatory bodies that are going to enforce that, right? And then how do you adjudicate whether or not someone's broken those laws and what the consequences are? There haven't been a whole lot of examples for us to really point to. Hopefully that means it's not really happening very much, right? But the first step is these kinds of bills, and we're working toward that. But I think more work remains to be done in this whole push to expand GINA to cover other kinds of agencies that might access your information is ongoing right now. And hopefully it'll be heartening for you to hear that it's actually geneticists, practicing geneticists, who are leading the charge on that.